Welcome back. If you haven't watched the first video, which sets up the question and provides some background on bacteria and these structures or fossils in general, make sure to do that before moving on here. So this video discusses 3 different tests that can be done to examine whether these structures are really microfossils. And these tests are, did the sediments form on the seafloor? Are the structures made of cells? And are they composed of organic carbon compounds? So to answer that first question, more detailed geological studies at the site later on have shown that the layer of chert rock actually cuts diagonally up through the layers of volcanic lava rather than forming on the seafloor. The diagram on the right is a map of the different rocks, as if you're looking straight down from above. The gray areas are the lava layers that formed on the seafloor. There's a dark gray area that also shows chert that formed in flat layers on the seafloor, but the fossil actually comes from the chert that cuts through the lava and not the chert that formed on the seafloor. In fact, the chert layer that the fossils came from was interpreted later on as a vent within the ocean floor, part of a hydrothermal system where superhot water comes up through cracks in ocean floor rocks. We know of similar hydrothermal vents in the deep ocean today. So what do you think this means for the hypothesis that these things are fossils if they formed within the seafloor at a hydrothermal vent? Well, sometimes the bacteria do live around hydrothermal vent systems, so we can't rule out the specimens being fossils. But this observation does mean that they likely can't be cyanobacteria. And see if you can figure out why that might be. Well, if the bacteria are living within the ocean floor, there would be no sunlight, and we know cyanobacteria need sunlight for the process of photosynthesis, so that rules out them being cyanobacteria. So next up, are the structures made of cells, like you would expect if they really were bacteria? This was the first question that was raised about the interpretations of the structures as bacteria in 2002, about a decade after they were first published. Some of the structures end up branching, which you wouldn't expect in bacteria, and sometimes the thickness changes considerably within one structure, which you really wouldn't expect in bacteria. But you could argue, and it has been argued, that perhaps some of these messy-looking specimens have just decayed a bit before they fossilized, or there are a couple individuals that got squished together as the rock formed and they decayed, or something like that. After making the observation that many specimens were a lot more irregular than would be expected if they were bacteria, this new research group proposed a different hypothesis, that the structures are not bacteria, but are just blobs of carbon that stuck to the edge of crystals. So in this photo, the arrow points to a crystal edge that's vertical and then bends sharply to the right, which is actually away from us into the rock. The carbon filament in the box seems to follow that bend. And interestingly, part of this specimen, before it bends, was one of the original species described and named as a bacteria. However, in some specimens, they don't obviously line up with the edge of crystals, and the original research group went back and did some more detailed analysis of the structures. They mapped the distribution of carbon in 3 dimensions, and they showed that they were spherical chambers, kind of like you'd expect from bacterial cells, at least in some of the specimens. So maybe there is evidence for bacteria after all. But that wasn't the end of the debate, and not by a long shot. The back and forth has continued even until today, including this study that looked at another of the original specimens. They also mapped the distribution of carbon and showed that yes, the specimen is indeed made of carbon, which is the bright colors in the central photo there, the bright orange colors. But in addition, they mapped the distribution and the orientation of the crystals. So crystals, in many cases, have like a long direction and a short direction, and it's possible to map out where those directions are pointing. And that's what's shown in the right-hand figure by the bright colors. And you might notice that the colors on opposite sides of the specimen, which is the black line in the middle here, seem like they line up pretty well. The crystal at A, which is the red and orange sort of blob there, seems like it looks like a continuation of crystal B, and so forth. You know, C and D seem like they match up, E and F. It almost looks like you could push both sides back together like a jigsaw puzzle and have it match up. In fact, you could interpret this as originally continuous crystals that were then separated by a crack that opened up and split them apart. So there is abundant carbon in this specimen, but it's possible and it seems likely that the carbon came in later and filled in a crack that opened up in the originally solid rock. And this wouldn't be consistent with the rock forming around an existing bacteria fossil and encasing it. Okay, so the structures are pretty hotly debated, but we've seen and you've heard that they do contain carbon. However, carbon can come in many forms, like the graphite that makes up pencil lead or diamond, and not all of those forms are created by living organisms. So we want to know, is the carbon in these structures the kind of organic compounds that you would associate with life? One of the ways that the carbon compounds have been investigated is with a technique called Raman spectroscopy. Basically, it shoots lasers at a substance, and different types of chemical bonds between atoms produce different peaks in the spectrum. In particular, there's two peaks that are associated with chemical bonds between carbon atoms, these D and G bands in this wiggly line. And so the diagram on the right compares these D and G bands in the Apex Chert, these sort of debated ones from 3.5 billion years ago, with carbon from younger bacteria fossils. In fact, the 800 million year ones from the Bitter Springs are the ones that I showed in the first video as the well-preserved bacteria. So these peaks are generally pretty similar, and it suggests that all of these materials, both the debated Apex Chert and the better accepted younger fossils, have similar types of carbon. But does carbon indicate life? The Raman spectroscopy just shows the types of chemical bonds, so we know there's chemical bonds between carbon atoms, but it doesn't prove anything about the origin. In fact, there are simple organic carbon compounds with those types of bonds on Mars, on Saturn's moon Titan, even in space, but there's no evidence yet, at least, for life in those places. And in fact, there is a known method called Fischer-Tropsch processes that combine inorganic compounds, like carbon monoxide, CO, and hydrogen, and do a bunch of chemical reactions, and end up producing organic carbon compounds, these chains of carbon molecules stuck together with hydrogen sticking off the side. It's also possible to produce organic carbon compounds from the interaction between salty water and minerals in rocks that turn carbon dioxide into organic compounds. So, although these methods aren't producing a lot of carbon on Earth today, when there's tons of life around, maybe they could have been more important before there was life. Well, I'll give the last word here about the carbon to Bill Schopf, who originally described the fossils. In 2018, he and colleagues measured the composition of the carbon molecules, something called the carbon isotopes. And I won't explain isotopes here in much detail because you'll cover them in the next topic. But they're basically the amount of different types of carbon in a molecule. And so he argued that the different species of bacteria in the Apex Chert tended to have different values of carbon isotopes. And so if the carbon was just random stuff that sticks to the side of cracks or the sides of crystals, you wouldn't expect the different shapes to have consistently different values. You might expect it to be just kind of random. But if they were really bacteria that use sunlight for energy should have different carbon isotopes from ones that use methane for energy, for example. And so if there really are consistent differences between the different shapes of fossils, that could support a biological origin after all. Okay, well that's the end of the story for now, but this is an area that still has a lot of controversy and it's probably not the end of the story forever. So what do you think? For the three questions, do you think that the evidence points more towards the structures being real microfossils of bacteria or inorganic features that aren't evidence for life? Thanks for listening.